So every vertebrate has mucosal barriers which are in direct contact with the environment. In the case of terrestrial vertebrates, our mucosal barriers are the lungs, the nose, the gut. But early vertebrates like teleost fish also have mucosal barriers. In the case of teleost fish, there's some that are in common with us, so the gut is a good example, right? Both fish and us will have a gut, but there also have some unique ones. The most obvious ones is obviously the gills. You can think about the gills being unique to fish, and that's a mucosal barrier, and also they have a mucosal immune system there. Because one thing that we do in my lab is to study the immune responses in the skin, so for that we basically just use this sterile glass slide, and we scrape the mucus up and down. And by doing this, we collect both the mucus secreted by the fish and also the bacteria, the good bacteria that live. Okay, so then the next thing that we do is actually we bleed the fish so that we get rid of all of the blood, and we also collect the plasma later on from this. So we bleed from the caudal vein. So we stick the needle in there until we reach the backbone. The next thing we're going to do is to dissect the fish. And we collect a number of organs usually, so we try to utilize the fish as much as possible for different types of experiments. So we create our initial dissection window where I open the peritoneal cavity. And you can see here all of the organs. I can So one of the things that we are going to collect today is the spleen. So if you look at the peritoneal cavity of the trout, this is the gut. You can see that the fish has eaten a little bit. That's the stomach over there. That's just fat. And that's the spleen here, this dark organ. Cut it out. And then we're going to place it in our tube here. Discovery was that we, for the first time, characterized the presence of an immune system associated with the nose of teleost fish. So as you can see, teleost fish have two nares, one here on the left and one here on the right, and the water goes through there, and that's how actually fish smell. So they will be detecting waterborne odorants through that water that is passing through those nares. And the olfactory organ is going to be a rosette looking, so almost like a flower looking epithelium that has the olfactory neurons, and as we have discovered, and additionally on top of olfactory neurons, it has a lot of immune cells that defend the trout against any dangerous organism that would be sensed through the nose. And also it's connected directly through to the brain, which is laying here. So to collect the nose, what we have to do first is to actually peel off the skin that covers the operculum, because otherwise we would get some contamination from the skin. So after we peel it off, then it's very easy to see where the two rosettes lay. So one there and one there. And then basically with the forceps, we just dig it down there and we pull the rosette up. And that's our tissue, and this has been what we have been using to do all of our experiments for this article that we just published. So no, that goes in there. No. So I'm just going to repeat the same thing with the other side. And it was very important that we bleed the fish first.